Alright guys, welcome to another video, and in this lesson I plan to show you how we can extract some DNA from some strawberries. But before we get into the experiment, let's try and understand exactly what DNA is. Well, DNA is found in the nucleus of all eukaryotic cells in these thread-like structures that we call chromosomes. And as humans, we have 46 chromosomes in each body cell. These chromosomes are found as pairs because we get one from our mother and we get one from our father. And because these chromosomes exist in pairs, our body cells are known as diploid. Now, DNA contains the genetic information that codes for all of our characteristics. It's what makes me, me, and makes you, you. Now, DNA is a long molecule made up of much smaller units. These units we call nucleotides, and each nucleotide has three specific parts. It has a phosphate group, we have a sugar, and we also have a base. And there are four possible bases that these nucleotides could have. They could have A, T, C, or G. And it's the ordering of these bases in our DNA that are specific to each human. Now, DNA is a double helix, meaning it's made of two strands of nucleotides. And these two strands of nucleotides are kind of wound together in this helix shape. For the DNA strands to join together though, we have to follow something called the base pair rule. And what that means is that A likes to pair with T and C likes to pair with G. Now, because DNA is specific to each of us, we can use DNA to find out who the father of a child is, for example, in a paternity test. But we can also use DNA as kind of like a fingerprint. For example, if there's suspects involved in a crime, we can take the DNA from a crime scene and that allows us to eliminate the suspects. So let's get into how we actually extract DNA from some strawberries. To extract DNA from strawberries, you just need a few easy to get hold of chemicals. Here I've got some isopropyl alcohol, also known as propan 2-ol, which was bought off eBay. During the packaging and delivery though, I think some of the isopropyl alcohol has escaped because it's dissolved the ink on the labels. You will need some washing up liquid. You will need your fruit. I found that strawberries and kiwis work the best. We've got our filter funnel. We've got some filter paper, again bought off eBay, and we have some table salt. Next, I took a plastic bag. You can use a sandwich bag or a freezer bag, and place the fruit inside the bag. Next comes the fun part, because what you want to do now is take your fruit that's in the bag and you want to break all of those cells up. So you just need to squash and squish that fruit as much as you can. The more that you squish the fruit up, the more cells that you're breaking open, which means the more DNA you can extract. Once you've squished up your fruit, about a couple of minutes, you then empty it all out into a measuring jug. Try and not tip any of it onto the table. Next, you need to make a solution of salt and washing up liquid. I'm just going to add a bit of distilled water. And you want a few teaspoons of salt. Now the salt, we're adding salt to actually break up any proteins that may be found in these cells. So we're just going to add a few teaspoons. And then finally, we're going to add some of our washing up liquid. Now, the washing up liquid is quite important because the cell membrane that contains the nucleus and all of that DNA is actually a fatty layer. The correct term we call it is a phospholipid. And the washing up liquid can actually emulsify or break down that cell membrane. Now, we want to add some washing up liquid, not to get a bubbly solution, but just enough to break open the cell membrane. And then we use a spoon to stir it all together. And now we can add our soap and salt solution to our crushed up strawberries. And I'm going to leave it to stand for about 10 to 15 minutes. Alright guys, so as I was filtering with the coffee filter, it was just taking way too long. So I opted to get a sieve from the kitchen. I passed the rest of the mix through and you can see that we've now filtered our strawberry mixture. Okay guys, so once you've filtered your strawberry mixture, you should have something that looks like this. It's quite goopy. It's got the consistency of like snot really. Looks a bit snotty. But we can't see our DNA yet because DNA is soluble in water. Now, if you remember the isopropyl alcohol that we had at the beginning, I put this in the fridge just to chill it. And when we add this to the water, as I said, that the DNA is soluble in water, but it's insoluble in our alcohol. So you just have to add it slowly. You just need to add your alcohol slowly to the side. And we should start to see the DNA precipitate out at the top. But we'll watch it all happen. There you go guys. The alcohol and the water form two separate layers. You can see that the alcohol is less dense, so it floats on the top of the water. But you can start to see, look, that our DNA is forming. The strands are all clumping together in the alcohol. And we're going to take it out with a spoon in just a second. So look, hopefully you guys can see here, look, that we've managed to extract some of our DNA, and the DNA is just this white stringy stuff that's kind of floating at the top here. So I'm just going to use a spoon and try and get some of this DNA out. And we're going to put it on a, there you go, look, lovely. Just going to try and scoop some of it out. And there you have it. There you go. We've just extracted the DNA from a strawberry. Alright guys, so I've literally just washed the DNA in some distilled water. It's there on my thumb. I'm gonna taste it. I'm gonna do it. Yeah, it just tastes gummy. It doesn't really taste... It just tastes weird. Alright guys, till next time.